Well, welcome back everybody to our last presentation for this semester. We are covering chapter 12 today, and this chapter is all about DNA technology. So this is looking at the cutting edge use of our knowledge of DNA for many purposes. We're going to look at genetic engineering, we're going to look at medical technology, we're going to look at forensic science, we're going to look at gene therapy, which tries to fix genetic problems, particularly in humans, but it's also done experimentally in other species. And at the very end, we'll talk about some ethical issues that surround the use of DNA technology, because they're also important considerations. And it is very likely that at some point in your lifetime, you will probably have to make some decisions about the utilization of DNA technology relating to you personally, for reasons which I'll get into when we talk about the ethics of the use of DNA technology. So let's begin at the beginning. One of the things that has become very common in forensic science and police work is the use of DNA fingerprinting and DNA profiling. And this has become way more sensitive than it has been over when it was first utilized. When we first started using DNA profiling, you needed a substantial amount of biological material to act as a source of DNA in order to be able to generate a DNA profile. But as you'll see as we dig into the use of DNA technology in forensic science a little bit later in the presentation, you need remarkably little biological material these days. As little as 15 cells will provide enough genetic material to do a DNA profile. Not because there's sufficient DNA in those 15 cells to do the profile, but because of what you can do with the DNA that you can get out of those 15 cells. And this is the reason why in the past, it has been very useful for police forces when they have reopened cold cases to have access to old biological material that had been stored. So long as it was stored in a way that preserved the DNA, forensic scientists can revisit that material, and using modern technology that allows you to utilize much smaller biological samples, you can do DNA profiles and therefore perhaps identify the perpetrator of a crime from many years ago. And you can see here that there's a reference to the Boston Strangler. There's actually a more detailed examination of that case right at the beginning of the chapter. But there have been many cases over the years, and every year you see a piece in the news, and it comes up quite frequently, of an old case being solved by the use of DNA technology. And when we talk about potentially doing your own personal DNA profile, we'll revisit this, because in some cases, the police have actually turned to publicly available DNA databases in the form of Ancestry.com and a couple of other places. I'm not sure about 23andMe, but I know Ancestry.com has been used, and they have actually compared some of their sequences against these publicly available DNA databases that people have, where people have voluntarily provided their DNA for sequencing purposes. And just so that you know, I've actually had my genome sequenced twice, once by the National Geographic Society as part of a research program that they were funding, looking at human migrations over time. That has subsequently shut down. They actually shut it down at the end of last year. So you can no longer contribute to that research program. But I've also had it done by 23andMe. And occasionally I agree to let them use my stored DNA sequence, because they have it on record, as part of their ongoing research into human diseases and the correlation between genetic sequences and human diseases. But they always ask for my permission And so when they ask, I consider whether I want to contribute to that study. If I do, I say yes. If I don't answer, and they won't use my sequence. So yeah, you can get your personal DNA sequence, and it doesn't cost very much. And that is one of the attractions to using it, but it also is, as you'll see, one of the reasons why we should pause and perhaps consider under what circumstances we want to have this done. But more of that later in the presentation when we get more into the ethics and we get more into the personal DNA sequencing story. So there are many things that we can do with our current knowledge of DNA and our ability to be able to manipulate it. Of course, you have heard of the use of genetic engineering to genetically modify organisms. Plants are the ones that probably get the most notification, but as you'll see, as we proceed through this presentation, a whole host of organisms have been genetically modified. And I might add that genetic modification of organisms is not something that is actually new. It's the way we go about modifying them that is new. Humans have been genetically modifying organisms for the best part of 12 to 18,000 years, ever since we started to become agriculturalists. The moment we take a wild plant and start selectively breeding it for the particular characteristics that we want, we are genetically modifying them. We're changing its genome. We are selectively preserving certain genetic sequences that yield the features that we want, and we are slowly but surely weeding out the genetic sequences that produce the features that we don't want. That is genetic modification, however you want to look at it. So when you look at all the breeds of dogs that are out there, we've genetically modified the original dog ancestor, probably a wolf, to give rise to all of the breeds of dogs we have. And we can make the same statement about any organism, plant or animal, that we have domesticated. What has changed is the way we go about genetically modifying them, because rather than breeding them selectively and hoping that we can preserve the features that we want and eliminate the ones that we don't want, we now can actually identify the individual genes in an organism that possesses the features that we want. And because of the commonality of the genetic code, we can transfer those genes into another organism that we would like to see that gene in. And that gene will be recognized by the new organism and expressed as if it was in its original host. That's the difference between our old form of genetic modification, selective breeding, and what we're doing now. So the reality is that when we start talking about genetic modification, we have to be very careful about arguing that somehow what we're doing now could be more dangerous than what we've done in the past. That may not necessarily be the case. And more of that as we proceed into a discussion of genetic modification. We've also been able to look at the genomes of different species. Many species that had small genomes had their genomes sequenced using the old sequencing technology, which was very laborious. When I first started in the molecular biology area of biology many years ago and started doing a little bit of gene sequencing myself, we felt that we were doing really well if we were able to sequence somewhere between 500 and 1,000 base pairs in a month or two. It was very, very labor-intensive technology to utilize. Today, we can sequence a complete human genome, about 3 billion nucleotides or thereabouts, in a day or two, and do it for under $100. That's the huge leap that's been made in sequencing. And as a consequence of this, we are now able to use sequences of genomes and do comparisons between genes and learn a lot about the evolution of different species, the relatedness of different species, and also to be able to get some information about how species have interacted with each other over time. So when our own species, Homo sapiens, migrated out of Africa somewhere around 60 to 80,000 years ago, probably closer to 60,000 years, but certainly no further back than 80,000 years. They encountered other human species as they migrated, first through what is now Palestine, then up into Europe and across into Asia. They encountered other species that we would have to consider to be human. You may have heard of some of them. So there are the Neanderthals, who we are very closely related to. And we'll talk a little bit more about that when we talk about personal sequencing. Also, there is a group that are known from just a few fossils from a cave in Siberia called the Denisovans. And we know that they have had an impact on modern humans because DNA from them is found in the genome of people that we now refer to as Melanesians and Polynesians that live in the Pacific. So almost certainly the ancestors of Melanesians and Polynesians encountered the Denisovans, and they obviously had sex with each other because there is the presence of Denisovan DNA in the genome of Polynesians and Melanesians. We could talk about a couple of others, other unique human species that we may or may not have interacted with. We're not entirely sure, but we know that they existed on the basis of the fossil evidence, and our ability now to be able to extract intact DNA very carefully and under very, very heavily controlled conditions to make sure that we don't contaminate the sources, and be able to take a look at their DNA and compare it with their own. So it's been a very useful tool in a number of different ways for biologists. And then in medicine, we are starting to apply what is known as gene therapy to fixing human diseases by introducing normal or unmutated normal versions of genes into individuals who are carrying potentially fatal genetic diseases. We've even got to the point now where we are starting to reverse some forms of blindness for people who have lost the ability to see because of genetic breakdown of the retina. This technology was actually pioneered in golden retrievers because there is unfortunately a genetic mutation in golden retrievers that leads to the same loss of eyesight. And the technology was pioneered in golden retrievers and very successfully restored the eyesight of a number of dogs that established proof of concept. And that same technology has now been used experimentally in humans very recently. And so the jury is still out on just how successful this treatment is going to turn out to be, but it's hoped that it will be just as successful as it was in the golden retriever situation. And there are many other examples that I could quote, but we will talk a little bit more about the use of DNA technology in medicine when we get there. So that gives you some ideas of some of the things that we have been able to do with our use of DNA technology. And we'll get into all of those in some detail as we progress this morning in this presentation. So when we use the term biotechnology, it's really all about the manipulation of organisms. And as I said, we've been manipulating organisms since we began the process of agriculture. We think about 18,000 years ago, certainly by 12,000 years ago, we know that humans were practicing agriculture. They had established settlements. I hesitate to call them towns and cities, but they were certainly collections of people that were able to live together because they could grow larger quantities of food, because they had domesticated plants and they were growing those and harvesting the products. They had domesticated cattle. They started to domesticate well, the dog was the first animal to be domesticated, probably 18,000 years ago, or maybe even a little bit later back in time. But certainly by 12,000 years ago, dogs were a common part of the settlements, as were cattle. I think sheep were probably domesticated at that point, but certainly the ability to domesticate organisms, to selectively breed them, to create organisms that were useful for our own purposes, allowed us to settle down and to practice agriculture. Instead of being hunter-gatherers, having to migrate periodically to new locations because we'd exhausted all the food resources in a particular location. Yes, that triggered the mass migrations of humans around the globe, so that by around 40,000 years ago, there is evidence of the ancestors of modern Aboriginal in Australia at that point. So bear in mind that the ancestors of all humans outside of Africa started migrating out of Africa as recently as 60,000 years ago, no later than, or no earlier than 80,000 years ago. And then by 40,000 years ago, they're already starting to invade Australia and having to cross an ocean or a sea strait in order to get there. So that means that there was obviously a strong force forcing people to migrate and migrate over reasonable distances in short periods of time. It's estimated that probably people were migrating on average about 10 to 12 miles per year as a consequence of this food pressure and the exhaustion of local supplies. They're probably moving about 10 to 12 miles. And it turns out that that's fast enough for people to migrate all around the edge of the Middle East and Asia and down into the Indonesian archipelago to be invading Australia about 40,000 years ago. So the math works. But we know all about that again from our use of DNA technology. And again, more of that a little bit later. This is a chapter where I could spend hours talking about this. No, I'm not going to spend hours talking about it, so don't panic. But yeah, we are going to spend a couple of hours probably going through this presentation because there's so much information in here. And there's so much more that I'd love to include, but simply can't because of the necessity to keep this short enough that you don't lose interest too quickly. Yeah, let's just move on. So, as I mentioned in my introduction, the big deal now with this whole genetic manipulation is the fact that we can now actually introduce individual genes through our ability to manipulate DNA. And this leads us to this notion of genetically modified organisms, or GMOs. Any organism that has a gene that's been introduced from another organism is referred to as transgenic. So that's another terminology that you'll hear me use, the term transgenic, which I think, well, there we go, the transcription service finally picked up transgenic. So this is another term that you're going to hear me use as we progress through this presentation. And it can be applied to any type of organism. So there are transgenic bacteria, plants, animals. We don't manipulate fungi too much. And the reason why is because fungi are really chemically interesting in their own right. And we don't know very much about fungi just yet, but we're learning a whole lot more about them. And I wouldn't be surprised if at some point in the future, there are going to be individuals who are going to start genetically modifying fungi. But right now, I'm not aware of anyone that's really trying to genetically modify mushrooms and the like. But I suspect that it'll happen at some point. So the secret to doing genetic manipulation is to create recombinant DNA. That is, to introduce DNA from one species into the DNA of another species. Now, typically when we do genetic engineering, when we create recombinant DNA, we don't move the genetic sequence directly from one species into the second species. There are typically intermediate steps that need to be undertaken in order to be able to do this. And I'm going to show you several different ways in which we can create recombinant DNA in order to manipulate different types of organisms by introducing foreign genes. So here's an example of a genetically modified organism. If you look at this the fish in this picture, you can see that they have a yellow-green glow about them. And that's because they are expressing a protein called green fluorescent protein, otherwise known as GFP. It comes from a deep water jellyfish, and it allows the jellyfish to glow in the deep ocean where it lives. Now, the reason why the jellyfish wants to glow is for two reasons. Number one, it attracts food to it because it's the only light source in the neighborhood, so that attracts small organisms, small fish and other things that it can eat. It also might attract other jellyfish to its location. Now, jellyfish do not mate with each other, but they do release their gametes into the ocean. So being in the same neighborhood as each other when they release the gametes increases the likelihood that those gametes will find each other in the ocean and therefore fertilize each other to lead to the next generation of jellyfish. Of course, there is a downside to all of this because if you are the only thing that's glowing in the dark, you also attract predators. So it's a little bit of a two-edged sword for the jellyfish. But for genetic engineers, people who want to genetically modify organisms, this protein and two others that we've extracted from deep water jellyfish, called very imaginatively red fluorescent protein and yellow fluorescent protein, so you can imagine what color they are. We have the genes for those and we can use those as marker genes, and that's why these fish actually glow, because the gene was not inserted into the fish just to make them glow. The gene was actually used as a marker. It was inserted into the piece of DNA that carried the genes that the genetic engineers really wanted to insert into the fish. And the way they can tell that at least some of the DNA, if not all of the DNA, got into the fish is the fact that the fish start glowing. Any fish that doesn't glow didn't get the piece of DNA that carried the genes of interest, and therefore it doesn't glow. So all of these fish are glowing because the marker gene, the gene for the GFP protein, got in. And this is the reason why we like these proteins. In the past, when I think back to when I first started with genetic modification of bacteria, we used antibiotic resistance. Yes, I know that sounds really silly, a really silly thing to do, because our big concern about bacteria right now is the fact that they are becoming increasingly antibiotic resistant. So why would we want to use antibiotic resistance as a marker for genetic engineering? Because it was the only thing we had at the time. Back in those days, genetic antibiotic resistance was not considered to be an issue. Because this is many years ago now, over 30 years ago. And significant antibiotic resistance among bacteria was just something that was not heard of. And we used the antibiotic resistance genes as markers, knowing that we were creating antibiotic resistant bacteria, but we always kept everything that we created and put it into autoclaves, which are sterilizers, and heated everything up to above the boiling point of water, above 212 degrees Fahrenheit or 100 degrees Celsius, and held it there in a steam-containing environment for 15 minutes to kill absolutely everything before we disposed of it. And that way we could guarantee that we were not releasing antibiotic resistant bacteria into the environment, because that would not be a smart thing to do. But it was also realized that using those genes as markers would in the end not be a smart thing to do. And so different systems were looked at. And along the way, we used some genes that we've talked about before, the lac operon, that's there to digest lactose when it's present in the environment. Well, it turns out that you can also use an artificial substrate to activate the lactose operon or the lac operon. And when it gets processed, that artificial substrate actually changes color from white to blue. And so you can detect the presence of an active lac operon, which is what we were doing, and we were using as a marker by using this artificial substrate. Then eventually we stumbled across these fluorescent proteins. First the GFP, and then YFP and RFP, yellow and red fluorescent protein. And we now use those as markers. So that's why these fish glow green. That gives you a little bit of more information about the basis for doing genetic engineering. But now we need to talk about the actual process. So it begins with the recognition that bacteria have not only genomic DNA, and here you're looking at an exploded E. coli cell that has been ruptured and its genome has erupted out of the cell. So that's this mess of squiggly material. This is actually the genomic material of the bacterium. But if you look very carefully, you can see that there's a smaller circular molecule here. There's actually another one here. There's another one here. These are independent of the genomic DNA. And these are referred to as plasmids. These are auxiliary DNA molecules, and they have genes on them as well. And what is really interesting about these plasmids is that bacteria can take these up from the environment. So if these plasmids are loose in the environment, bacteria have the natural ability to take these plasmids up and therefore take up the genes that are in these plasmids. This is in fact the way a lot of bacteria become antibiotic resistant, because some antibiotic resistance genes actually live on these plasmids. They're called R plasmids, for resistance plasmids. And if a bacterium that has these plasmids, these resistance plasmids in it, releases some of those into the environment, other bacteria can take them up and do so relatively easily. Now, and that's how they become antibiotic resistant. Now, we can utilize this ability of bacteria to take up plasmids to introduce our own genes. Initially, we used plasmids derived from bacteria and learned how to manipulate those and learned how to introduce genes into naturally existing plasmids from bacteria. But eventually, with the ability to manipulate DNA, we actually started to create our own versions of plasmids that could be taken up by bacteria. And we started to engineer the genetic sequences of plasmids to make it easier to insert foreign genes into the plasmids in order to get them inserted into bacteria. And we also developed techniques for encouraging the bacteria to take up the plasmids. So the simplest way to get a bacterium to take up a plasmid these days is to use a process called heat shocking. And I actually worked in the lab of a Japanese researcher who actually developed what is probably the simplest heat shocking process that we have at the moment for getting bacteria to take up plasmids. And basically what you do is you have a solution of bacteria in a small capped test tube. These are specially made for the purpose. You add a solution that contains the bacterial plasmids. So now you've got the plasmids mixed in with the bacteria. If you let them sit like that for long enough, some of the bacteria would actually take up the plasmids. But you can make more of the bacteria take up the plasmids by loosening the membrane of the bacterium. And the way you do that is simply raise the temperature on them. So you heat them to 42 degrees. You actually place them in a water bath at 42 degrees, and you only need to leave them there in this technique for 30 seconds. The old technique you used to have to leave them there for up to 5 minutes. But with this new quick technique, you leave them in the water bath for 30 seconds, and then you plunge them into ice to cool it down quickly again, to tighten the membrane back up again. And in that 30 seconds with the membrane open with sufficient concentration of plasmids, some of the plasmids will sneak into the bacteria, and the vast majority of cells will pick up at least one of those plasmids. Then you take the bacterial cells and you spread them out on agar plates. These are the plates that we grow bacteria on, that contain what is referred to as a selective medium. It contains something that will select for the bacteria that have taken up the plasmids, because of course in the plasmid is one of our marker genes. Now with the green fluorescent protein as the marker, you don't even have to put anything on the media in the agar plate to know whether it's been taken up or not. You just look for the bacteria that are glowing green. And by spreading them out sufficiently, you can spread out the bacteria so you've got individual cells spread out over the surface, and each cell will then grow and divide, and create a little colony of cells all derived from that original parental bacterial cell. And that's what you're looking for. You're looking for these tiny little colonies of bacteria that are glowing green. They're the ones that have got the plasmid in them. So how do we go about introducing genes into the plasmid if the plasmid's the secret for getting the genes into the bacterium? Well, this revolves around using a technology, and this diagram just simply goes through the technique that I told you about. It's not going to, the idea of putting DNA into the plasmid and then getting the plasmid taken up by the bacterium, and then the bacterium produces whatever proteins you've got coded in the plasmid. And this just gives you a list of some of the genes that have been inserted into bacteria so that they can make a protein. So in the case of pest resistance, it's actually using a gene that's taken from a bacterium itself that is inserted into a bacterium that will infest the corn, called Bacillus thuringiensis. And Bacillus thuringiensis actually is deadly to insects. Doesn't hurt the corn, but it hurts the insects that might want to feed on the corn. For cleaning up toxic waste, this is one that's still been heavily investigated, because our ability to genetic engineer bacteria to do this has not been terribly successful at the moment. So people are still working on that one. Insertion of insulin in the insulin gene into bacteria, that has been very successfully done. In fact, if you are a diabetic, you may very well be injecting recombinant insulin. It's called Humulin, or at least one brand of it is called Humulin. And I've got a picture of the bottle a little bit later. It may actually be the next slide. And that's because the alternative source of insulin is the pancreases of pigs from slaughterhouses. And they extract the insulin out of the pancreases of slaughtered pigs. And pig insulin is close enough to human insulin that most people can tolerate it. But if you have a bad reaction to it, because insulin is actually a protein, and it's proteins that the immune system react against. So if your immune system happens to react against insulin, the pig insulin, you could be in a bad way as a diabetic because you can't utilize the pig insulin. There is an alternative source, and that's bovine insulin or cow insulin from the same source, pancreases of slaughtered cows. But if you could inject straight human insulin, then there would be no problem because it would be recognized as a human protein, and hence the effort to clone the insulin gene into bacteria. It was one of the first ones to be cloned into bacteria, along with the genes for 2 proteins that are missing in individuals who suffer from, I'll remember what it is in a moment. I'm having a senior moment. We talked about it when we were talking about sex-linked diseases. And now I've gone blank. Hemophilia, there it is. I knew it would come back if I let it vegetate for a while. Hemophilia. So there are two genes, or 2 different genes that are affected in hemophilia. Depending on which type of hemophilia you have, determines which of the 2 genes are in effect, are turned off, and therefore you're not making the protein. And there are multiple proteins that are required for blood clotting. So if you're missing one of the proteins, and it's clotting factor number 8 and clotting factor number 9 that are affected here, if you're missing one of those 2 proteins, then your blood won't clot. And the only source of getting those proteins until the genes were cloned into bacteria was the human blood supply. And the problem with that was from about 1979 through to 1982 or thereabouts, the human blood supply was contaminated with HIV because there was no way of identifying blood donations that were contaminated with HIV. We were relying on people to be honest about their HIV status, and many people were, but some people weren't, and there was no way of specifically knowing for sure that the blood supply was not contaminated with HIV. And for hemophiliacs, the only choice they had was to get the blood clotting factor that was available and run the risk of becoming HIV positive. And that actually occurred for about 40% of the hemophiliac population here in the United States. They contracted HIV through contaminated blood clotting factor. That's not an issue anymore because the gene has been successfully cloned in bacteria, and the bacteria now make the 2 missing clotting factors, and the pharmaceutical companies simply extract that out of the bacteria, purify it and package it, and it's made available for hemophiliacs, along with the insulin and other proteins of interest. So this animation, which you can find on the resources page in Canvas for Chapter 12, so you need to go to the Chapter 12 resource page, simply walks you through the process of cloning the gene into the plasmid. It still doesn't tell you exactly how it's done. It just sort of leaves it as a little bit of a black box, but I'll explain to you specifically how we get the gene into the plasmid and guarantee that we get it in there in a moment. So how do we really get that piece of DNA of interest, the gene of interest, into the plasmid? How do we merge the 2 DNA molecules together? Because that's the key. Well, it comes back to bacteria again and a way that bacteria protect themselves from foreign DNA. Remember I said that bacteria can take up plasmids? Well, they can also take up other types of DNA as well. The plasmids are the ones that are useful to them, but they can take up foreign DNA of all sorts of types if it's in the environment. They can't pick and choose. It sort of happens because that's what they do. So if there's foreign DNA that they've picked up that's potentially dangerous to them because it has genes that they don't want that are dangerous to them, then they need to be able to protect themselves against that. And so they have enzymes called restriction enzymes. We discovered restriction enzymes back in the early 70s. Now we know of hundreds of these restriction enzymes. The first one was discovered in the bacterium E. coli, Escherichia coli, that is the bacterium that lives in our colon, hence the name coli, referring to the fact that it comes from our colon. And the enzyme that was extracted out of it was called EchoR1. And we actually use the ECHO-R1 as an example restriction enzyme in the next diagram. So the trick with restriction enzymes is that they recognize specific nucleotide sequences in the DNA, and they cut the DNA at those particular sequences. So in this case, this is the ECHO-R1. So this is the ECHO-R1 restriction sequence. That's the sequence of nucleotides that the ECHO-R1 enzyme actually recognizes. And you notice that it's symmetrical. It reads the same in each direction, and it cuts. And if I remember correctly, this is the way it does it. It cuts. Okay. Yeah, see, it's got it down here. If I'd looked a little bit lower, I would know. But that's where it cuts. And it creates these things, sticky ends. You notice that the sequence here is complementary to the sequence here. This is why they're called sticky ends. So the first thing that you do is take the DNA that contains the gene of interest and digest it with the restriction enzyme so that you create sticky ends on the end of the piece of DNA that you want to insert into the plasmid. Then you also treat the plasmid with the same enzyme, and there will be a restriction enzyme sequence, an ECHO-R1 sequence in this case, within the plasmid. And so now you've got a piece of DNA that has a sticky end that matches the sticky ends of the cut plasmid. So when you mix the piece of DNA that contains the genes of interest with the cut plasmid, these sticky ends will do what sticky ends do, stick to each other because the sequence of nucleotides is complementary to each other, they base pair. Now, there are still gaps here that need to be sealed, but if you remember from our discussion of DNA replication, we can seal those gaps using an enzyme called DNA ligase, because ligase recognizes the gaps and seals them. It does that during DNA replication. And you can buy DNA ligase. You can isolate it from bacteria. And you add it to this mixture. And the ligase seals the plasmid. And now you've got the plasmid with the inserted piece of DNA. And this is your recombinant DNA, your recombinant plasmid. That's what you then introduce into your bacteria solution. Do the heat shock. Bacteria take up the plasmid, and now you've got the plasmid with the gene or genes of interest inserted into the bacterial cell. Now, if your intention is to go on and into one of our health science programs, ultimately you're going to probably have to take our microbiology course. And in our microbiology course, you'll get an opportunity to actually do this. One of the experiments that is performed in our micro course is to introduce the GFP protein, the green fluorescent protein, into bacteria and to create bacterial colonies that glow green. And it's something that our microbiology students do every semester. This is a very, very easy technique to use. In fact, I know of people who do this at home on their kitchen bench, just as you can clone plants on your kitchen bench at home. We talked about that in the last presentation. So this type of genetic engineering can be quite easily done on the kitchen bench at home if you have the right conditions. There are high schools that use this in their biology classes. Maybe you went to a high school that had a really good biology class and you may have had an opportunity to do this, because I know biology students in high schools are doing this sort of thing these days. It's really easy to do. And it gives you the opportunity now to introduce this into a bacterium and have it express these foreign genes. So this animation just simply talks a little bit more about restriction enzymes. So here you have the restriction sequences of 3 different restriction enzymes. So this is our old friend ECOR1, the E. coli gene. I think this might be BAMH1, if I remember rightly. It's been a long time since I've looked at these sequences, and I haven't looked at this animation for a little while, so I'm running on the vagaries of my memory, but I think this might be the BAMH1 restriction sequence, because it also gives you sticky ends, just like the ECOR1 does. And then this is one that I'm not familiar with, but this one is another restriction enzyme, but notice this one does something a little bit different. Rather than creating sticky ends, it creates what we refer to as blunt ends, because it cuts at a particular sequence of nucleotides, but it cuts both strands in the same location rather than offset like this to create sticky ends. And sometimes you want these blunt ends. And so depending on what you're trying to do with your cloning, you may actually want to do blunt end cutting with a restriction enzyme. So again, you can find this animation in the resource section in Canvas, the Chapter 12 resource page. So you can take a look at this, and it will introduce you to these restriction sequences and what you can do with these restriction sequences. This, as I said, this is very, very commonly used technology these days. Then perhaps the most amazing technology that we have now that allows us to do in situ gene editing and even to introduce new genes is to use something called the CRISPR-Cas9 system. Now, this is also based on a system that exists in bacteria. We know that this is used by bacteria to disable viral DNA. You may recall, I think we talked about this in, and I'm trying to remember whether I talked about the Hershey-Chase experiment using bacteriophage in the previous presentation, in the DNA replication presentation. It's been a while since we did that. It's a couple of weeks ago. So I can't remember whether I talked about that. But one of the things that we know is that there are viruses that specialize in attacking different types of organisms. So there are viruses that attack mammals, some that are specialized in humans and other different types of mammals. We know that COVID virus came out of probably bats via some other intermediate to enter into humans. HIV came from chimpanzees, but originally came from monkeys via the chimpanzee to humans. And most viruses that affect humans have come from other animals, either directly or indirectly. Then we have viruses that attack plants, and no plant viruses won't attack humans. If the virus attacks a plant, it's a plant virus, not a plant virus, but perhaps something else. No, they only attack plants. And then there are viruses that attack bacteria, and these are called bacteriophages. And of course, for a bacterium being a single-celled organism, when a virus takes over a bacterial cell, bad things can happen because there are two different ways the virus can affect a bacterial cell. If it's a non-lytic virus, that means it doesn't rupture the cell. The virus particles emerge from the cell and the cell remains intact. So the bacterial cell is still intact, but it's still just a virus factory. It's not functioning as a bacterium anymore. But then there are lytic viruses, which means that when they want to emerge out of the cell, they burst the cell open to release the virus particles, and that means the death of the cell. Same thing happens with human viruses and plant viruses and all viruses, these two basic types, lytic and non-lytic. And so for a bacterium to be infected by a bacteriophage is bad news either way. So they need a way to cripple the virus, and the Cas9 system evolved in bacteria to do this. It contains a guide RNA molecule that complements the sequence of nucleotides in the viral sequence, and that guides the protein complex that's associated with the RNA molecule, which is the Cas9 complex, down to the viral DNA and identifies the matching sequence. It base pairs with it, and then the Cas9 protein cuts the DNA and cuts out nucleotides. The DNA repair system of the cell then fills in the missing pieces, but it adds random nucleotides. And that typically disables the gene that's been targeted, and that in turn disables the virus because it's targeting a particular gene that would allow the virus to be infective of the bacterium. The CRISPR-Cas9 system takes that one step further. Biologists started using the Cas9 system to knock genes out because it was easy to do. That's what it's doing in the bacterial system. They could use this to target genes in different types of cells that they were interested in to knock out genes of interest. That would remove that gene's function, and they could then tell what the gene did. They could also see what dominoes fell afterwards by taking out that gene, because typically if you knock out one gene, it's not just that gene's function that you lose, there could be other things that are lost as well. They've even used that as a way of, in mice at least, correcting a genetic disorder that is very similar to Duchenne's muscular dystrophy in humans. And they've been able to knock out the mutant gene that causes that form of muscular dystrophy in the mice and replace it with a normal version, and therefore cure the muscular dystrophy. And it's hoped that at some point in the future, given that we now have proof of concept in the mice, although we've also learnt the hard way that mice are not little humans and we are not big mice. So what works in mice may not necessarily work in humans, but at least we've got a proof of concept that suggests that it's possible. But after all, the concerns have been ironed out about using the technique. It may be possible to use not just Cas9, but the CRISPR-Cas9, which is an extension of the Cas9 system, which I'll talk about in a moment, to actually replace the muscular dystrophy, and particularly the Duchenne's muscular dystrophy allele that is found on the X chromosome and affects males that inherit that X chromosome from their mother. So what does CRISPR-Cas9 do? Well, CRISPR-Cas9 takes it one step further. So let's take a look at the diagram rather than just keep talking about it. So here's the Cas9 complex with its guide RNA. That comes down and binds to the sequence on the cell's DNA, and then the Cas9 protein actually cuts out the nucleotides in this region. And so you've disabled that gene. But you also include, as part of CRISPR-Cas9, a copy of the normal gene, and that sequence of that normal gene is then used as a template to repair the nucleotide sequence in the disabled gene. So now you have a corrected gene, you've taken out the mutation, and you have a normal copy of the gene. So this would then allow you to actually correct a non-functional gene or a misfunctioning gene by knocking out the known mutation and filling in that gap now using the nucleotides of a normal copy of the gene that you've also introduced, so that that can be used as a template by the repair mechanisms, the cell's repair enzymes, to actually fix and correct it. Now, the way this would be done in human situations is probably a little bit different from the way it's being done right now in the mouse models, where it's being used, where the proof of concept is being figured out. What we would be more likely to do here is to target cells using an infectious virus that is known to target the particular cells that we want to use the Cas9 system, the CRISPR-Cas9 system on. And this falls under the heading of gene therapy, which is something that we're going to talk about a little bit later in this chapter. So I don't want to go into too much detail right now, but suffice to say that researchers have now created infectious virus particles that actually incorporate the CRISPR-Cas9 gene sequences. And so that allows you to use the virus as a vector, as a way of carrying the CRISPR-Cas9 system to the particular cells that you want to target. So it's not just a random introduction of the CRISPR-Cas9 system and hoping that it hits the cells that you want to get. You can actually target the particular cells that you want to target using this infectious virus. And it's a non-lethal virus. So it's not going to hurt individuals that are given this treatment. It's just going to target the particular cells that they want to alter using the CRISPR-Cas9 system. So this gives you some indications of some of the things that have been done. I've talked about the concept of humulin. So here's humulin. This is Eli Lilly's version of recombinant human insulin. And that's the correct way to describe it. It is recombinant human insulin because you've created a recombinant DNA in the form of the bacterial plasmid that contains the insulin gene. And you've introduced that into the bacterium. And the bacterium has then produced the insulin protein that can be then isolated from the bacteria and packaged. So this just adds to that idea. And here is the production plant. These are big growth vessels, basically, incubators full of nutrient solution that the bacteria will thrive on. Each is then inoculated with bacteria that contain the humulin plasmid, and they grow in the media, and they produce humulin, the human insulin. When there are enough bacteria in the vat and they grow to huge quantities, then they take the solution out, filter out the bacteria, and then use detergents and other agents to solubilize the bacteria and solubilize the lipids and the proteins, and then they isolate out the proteins. And then they particularly isolate and purify the humulin. They have a technique that allows them to batch purify large quantities of the humulin and to screen out all the other proteins. And they're very, very careful, and they do a lot of testing as they follow the process through to make sure that the only protein that they're capturing in the end is the insulin protein, so that they can package purified recombinant human insulin. This is the same basic technique that they use for all of those proteins that have been cloned in bacteria. So the blood clotting factors and other proteins that we talked about earlier are all grown in a similar fashion. So here are some of the other proteins that are now, for medical purposes at least, grown in bacteria. So human growth hormone, which is used for the treatment of some juvenile conditions. If your body doesn't make sufficient growth hormone, then you can receive injections of human growth hormone while you're young, and you ought to be producing the growth hormone so that you develop normally. Several proteins that are involved in breaking down blood clots, so-called clot buster drugs, that are utilized in people who have suffered heart attacks and strokes, which is basically a blood clot in the heart or in the brain, respectively. If you can break that clot up within an hour of the onset of symptoms, what is referred to as the golden hour, you can actually restore blood flow and minimize the effect. In some cases, depending on how quickly you get the the clotting factors or these clot busting proteins, you may actually prevent any damage at all, as long as you restore blood flow quickly enough. You do need to be very careful in the use of these clot busters, because as they are restoring blood flow, if the blood vessels have been damaged as part of this, then blood can actually leak out, and the bleeding into the heart, or if it's a stroke, the bleeding into the brain particularly, can have very bad outcomes. Without going into a lot of details, my family's had personal experience with this in the past. Several vaccines have been produced in bacteria. In fact, the recombinant vaccines that are produced by Pfizer and Moderna are produced this way, through genetically modified bacteria. It's one of the reasons why they were able to make them so quickly, because they were using techniques that were already well understood, and they simply adapted them to making the vaccines that have proved to be extremely successful against the COVID virus. And then erythropoietin, which goes by the abbreviation EPO, which was originally cloned because it's a hormone that encourages the production of red blood cells. And this was realized to be important for people who were going through chemotherapy and radiation treatment for cancer or any form of chemotherapy. Because unfortunately, chemotherapy particularly targets the cells that produce red blood cells. And it means that the individual can't make enough red blood cells and they become anemic. And that means they get weak, they lose their appetite, or because they're on treatment to save their life. But if you parallel the chemotherapy with erythropoietin, that encourages the body to produce extra red blood cells and offsets the effect of the chemotherapy. Unfortunately, some athletes have learnt that erythropoietin can do this and have been using this as a way of boosting the red blood cells in their bloodstream to increase their performance. This was one of the doping drugs that cyclists like Lance Armstrong were caught using. And this is the reason why Lance Armstrong was banned from cycling, because he was using things like erythropoietin to boost his performance. And part of the problem for sports regulators is to try and keep up with medical advances in this sort of area, where you can produce these performance enhancing chemicals for medical purposes, which athletes immediately latch onto and start using. And unless you've got a way of detecting it, which initially, most of the time you don't, it's impossible to know who's using these chemicals and who's not. Erythropoietin is infamous because of Lance Armstrong and because of that use. So let's take a break. We've been going for a little while and talking about a lot of interesting material. So let's take a break. But before we stop for the break, here are our concept check questions at this break, and I will meet you on the other side with the answers for these questions. Let me move this text box out of the way again. So here are the questions. Why are plasmids valuable tools for creating recombinant DNA? Think about where we've talked about them. A restriction enzyme cuts DNA at the sequence AATC, and this is an actual restriction enzyme sequence. How many cuts would this enzyme make in a section of DNA with the sequence CGAATC, TAGCAATC, GCGA? Come back and take a look at it after the break. And bacteria are easier to manipulate than yeast. Why are yeast used to produce some human proteins? Might have to think back a couple of chapters ago to find an answer to that one. Anyway, I'll see you on the other side of the break with the answers to these questions.